So under each version, or beside each version, there's, I think most of them have 6. 9 times 6 is 54, and that's 55 students in the class between the two sections. So what you want to do is look for your name, okay? And that's the one that you would print off. So here's one version. And so remember I told you I would have the answers on the side? So you can't answer it online, I don't think, but you need to go ahead and print it off anyway. But the colors are pictures, so if you want to refer back to the online version, if that helps you, or if you want to pay the money to print it. So in other words, you would, like I said previously, you would just mark the ones that are correct. And there may be some questions that have no correct answers, and you would get a quarter point per answer. So each question is worth a point. Okay, so there are 75 points total. Some questions just have 2 questions, like this one. And obviously only one, but you get a quarter point for not answering that one as well.
You said there may be questions that have no correct answer.
So if I say, aldosterone binds receptors on which of the following components of the nephron, and I might list structures that don't have any receptors. So that is, all of them could be right, one could be right, none of them could be right, and you would still get the quarter point per answer for knowing that that was an incorrect answer. Okay? So those are so here's some diagrams asking for respiratory patterns, compliance, and breathing patterns. So everybody has a couple of x-rays to identify. So again, this is easier to look at online and try to print it off and see the clarity. And then this one, similar to what I have on the board. But essentially what I've done here is just divided different parts of the nephron. And the red arrows mean it's coming out of the tube, and the green arrow means it's going into, so reabsorption and secretion. And then over here I list those substances, various types of diuretics and hormones. And so don't forget the chart, because I don't want you writing it in there. There's a chart here. So for the proximal convoluted tubule, from this list of things I've given, name 4 things that are reabsorbed, okay, or 4 things that are secreted. And then what type of diuretic is effective at that area, and any hormone that might work on the proximal convoluted tubule. Okay? If it's a black area, that means I'm not asking that particular question. Okay? So print this out, and it's due. I added a couple of extra days since I was late in getting it to you, so it's not due till May 3. It will obviously be in the digestive system by then, so you're welcome to turn it in any time that you are done with it. You don't have to wait till then, and that way I can get started grading it instead of having all of them on there. Yes?
That's both the respiratory.
Both respiratory here. First 40 questions are respiratory, and then the urinary after. There's some overlap with acid base. Okay? So you're welcome to work with each other. Like I said, the questions are similar. Some of them will be the same from one type of one exam to the other, but not necessarily all of them will be the same. So they're kind of merged. But some of them are really easy, and some you're going to have to do some research on. Okay? But the point is that you're going to have access to that, not have to memorize it, but still learn some information while you're taking it. Okay? Yes?
Does that mean that the acid-base extra credit is due on the 3rd also, or you want it on the 1st?
Let me think about that. Well, probably easier just to keep it to that date. That's what we'll be covering in lecture on Tuesday. Okay? So I just put these up at about 10:30 or so. Yeah, I know you can tell me when you first saw them, but I'm just teasing. She had them already printed off when she came to class. So they're available, and again, they're on the content page for that. Okay? Well, if you don't want to print it off, you can just write 1, 2, 3 on a piece of paper and circle it. Also, a good idea is to keep track, maybe on a separate piece of paper, your sources, so that you can say, but this is where I found this, or kind of a thing, rather than that's when I make the questions. That's what I was doing last night, was not only taking out the highlights and the answers, but I also had the email addresses of some of my sources in there, so who knows, I might have left a couple of them in there. But I was trying to catch those. So what I want to do today, before we move on to the function of the distal tubule, was to do a general review, look at the nephron loop again, all right? And then we'll look at the hormones that are regulating water movement in the distal tubule and convoluted tubule. We'll take a break, look at characteristics of urine because we're doing a urinalysis lab, and then we'll break for lab. Okay? So on the board, you have this diagram is very similar to what I have over there, but remember we have filtered particles that are small enough to come into the filtrate at the capsular space, and it comes in isoosmotic, or the same particle concentration to blood, okay, and to the interstitial fluid. This would be 300 milliosmoles around it as well. We don't start out with water being pulled out. We have to create the gradient, all right? And again, that's the purpose of the nephron loop, is to create that gradient, all right? And so the pump for that is in the ascending limb. So sodium is going to move out of the ascending limb, and as such, it's going to pull fluid with a higher concentration, and the fact that the descending limb is permeable to water is going to create an osmotic force that pulls water out of the descending limb. This becomes higher in concentration, and the reason we call this a countercurrent multiplier is that this higher concentration now moves over to the ascending limb, okay? And therefore can make the interstitial fluid even more concentrated, even more dense with particles. So that's what we were going through with the slides. And then the vasa recta function to take up that fluid both from the nephron loop and from the collecting duct. Okay? So at the nephron loop, it's also countercurrent with the nephron, with the vasa recta. So the nephron loop is going in this direction, the vasa recta is going in this direction while the descending limb is going in that direction, but it's not a multiplier because the two are not connected. Okay? So the multiplier is limited just to the nephron loop. So as salts are taken out and fluid is taken out, it can be taken up into the blood because this is an ongoing. We don't just create the gradient once and then leave it that way. We're constantly reforming it. And then the vasa recta can also take up the water. This is flowing parallel with the collecting ducts and take up any water if the channels in the collecting ducts have been opened. So let's look at this one more time, and then we'll move over to the chart. Usually I redraw it, but I couldn't remember, wasn't sure I'd be able to remember everything I put there, so that just came out as I was talking. I didn't have a form for it. So remember in the nephron loop, this is in the same sequence as we just talked about. What determines this osmolality, or the osmolarity of our filtrate? What would increase or decrease that osmolarity at this point, as we got to this point? In other words, is the filtrate in the capsule the same or higher than the filtrate in blood? Or lower?
The concentration?
The osmolality. So the primary ion involved in osmotic, now we also have proteins, okay, but the primary ion involved in osmolarity is sodium and chloride ions. All right, so the number of particles dissolved in the blood is going to be, our separation is purely by size. So as salt particles move across through the filtration membrane, water is going to follow passively. Right? And in the proximal convoluted tubule, where we're where we're pumping sodium out, chloride ions in water will also follow passively. Right? So we're going to maintain an equilibrium in that exchange. And we only have certain places where that doesn't take place. So that's why this is still the same as blood, okay, because there's an equilibration in that movement. So now we turn on the pumps, which are in the ascending limb. It seems backwards to start there. Somebody this morning said, well, why is it in the ascending limb? It's like, well, because this is what we're making concentrated. And the reason it's a multiplier is this higher concentration ends up over here. That creates the multiplying factor that we can get higher than 500 milliosmoles. All right, so what is moving the water across our channels here is that higher interstitial osmolality. And what creates that higher interstitial osmolality are the pumps in the ascending limb. Now I've drawn it in the middle just to show the effect here, but it would be all the way around. It would be the entire milieu of interstitial fluid. What is that?
Yeah, what is that?
That's my URL for the logarithmic calculator online. Use for acid base. I stuck it in there somewhere. So that's for Tuesday. So now we have the higher, so the descending limb is always going to be higher in osmotic concentration than the ascending limb. All right? And the interstitial fluid is going to be the same as the descending limb once those equilibrations have taken place. So now the descending limb fluid flows into the ascending limb. Hence the multiplication. This is what allows multiplication. All right? Otherwise it's just straight countercurrent. But the fact that descending fluid moves into ascending limb and becomes affected by those pumps is what creates our multiplication factor. Okay? And it's also the reason that the longer each of these tubules is, the higher this number becomes at the bottom.
And that's critically related to the impermeability of the ascending limb to water, correct?
Correct.
Yeah.
Otherwise, we would see the same thing in the proximal convoluted tubule. We'd have no change in concentration. We just have a passive movement. In fact, there's a question on one of the exams that asks about that. Or several of the exams. All right, so we see the same thing occurring. All right, so why do you think the tip of our loop has the highest concentration?
Being forced down.
All the water.
So notice the equilibration as sodium is being pumped out. What's happening to this level of sodium ions? It's decreasing. As it moves up, more sodium ions are leaving. As it moves up, more sodium ions are leaving. Okay, so it's just in the direction of the flow. Fewer sodium ions have left here, so this is the highest concentration. By the time it gets up here, it's more dilute. So therefore, the interstitial fluid that those ions are moving into is also going to be more dilute. So it works such that when our filtrate flows back down the collecting duct, it continues to increase in concentration in the environment around it so that we can, as need be, get to the higher concentration of the filtrate before it becomes urine. So finally we get to this final. And you can see that the filtrate leaving the ascending limb and moving into the distal convoluted tubule is hypoosmotic. It's below the number of particles per volume that blood is. So if nothing else happens, if we don't do anything to change this, your urine is going to be hypoosmotic. All right? So we have to actually do something different to change the osmolarity of the filtrate to make it a concentrated urine. So I know this looks a little busy. Some of you have already taken pictures of it, but let's kind of go through it slowly. All right? So we start our process of urine formation at the capsule. And this is what takes place at the glomerular capsule.
Are you planning on doing this drawing fresh, or should we
No.
draw it down right now?
Go ahead and draw it down. I would draw it fresh. I normally draw things fresh for you, but I have so many different things on here I was afraid I would forget. what they are. So the capsule is for filtration, and that's dependent primarily on size, but also charge. All right, some of the structures in the filtration membrane are affected by charge. Now, I don't have the capsule blocked off in that diagram I showed you on the exam, but the vessels going into and out of the capsule are affected by this hormone, atrial natriuretic peptide. All right, we've talked about this before as part of cardiac output, as part of blood pressure control. So what does atrial natriuretic peptide do? Is it going to increase blood volume or decrease blood volume?
Increase volume by decreasing salt.
What stimulates its release? Notice it has the word atrial in it. So what's stimulating its release? What triggers
The heart.
the heart? And is it not enough blood flowing to the heart, or is it too much blood flowing to the heart? Too much. So it's triggered by, I'm looking for my controls. It's triggered by a stretch of the atrial walls, which means that the purpose is to decrease blood volume. All right? So in thinking of it in that nature, and it's affecting the diameter of our arterioles, would you expect it to cause the afferent arteriole to constrict or to dilate?
Decrease blood pressure.
So we want to get rid of fluid. Right? Fluid is so individuals that have congestive heart failure are given a diuretic to increase their urine output and decrease blood volume. So as a hormone, atrial natriuretic peptide is going to lower blood volume by getting rid of sodium ions over there at the distal tubule that we'll get to later. But to increase blood volume, we need to increase filtration pressure, right? So to do so, it's going to dilate. Remember, the purpose is to make more urine. Okay, now I know it seems confusing because there are other substances that cause a dilation in the afferent arteriole was because pressure was too low, and we were trying, but it was too low for filtration, for the function of the kidney. All right, as it affects the kidney. Now we're trying to locally change the pressure so that we make more urine and get rid of the fluid. So it's not a systemic change. The purpose is to increase the urine output, okay? So now we move to the proximal convoluted tubule. That's what we talked about on Tuesday. And the role of the proximal convoluted tubule is primarily reabsorption. That's what the purple arrows are doing. This material leaving the convoluted tubule is reabsorption. Reabsorption meaning returning it back to the blood, not just absorption, like we do in the digestive tract, but reabsorption, taking it back to the blood. Okay? Secretion also occurs, primarily, remember, hydrogen ions and antibiotics. And we'll talk about that secretion process on Tuesday next week when we do acid-base balance. Okay. Throughout this flow, the osmolality of the filtrate is 300 milliosmoles. Okay, so we're not changing that, we're changing the content, but as we take particles out of the filtrate, water is leaving as well. So we're not changing the relationship, the dilution or concentration factor of that. Then our filtrate flows down the nephron loop and up the ascending. And here we have our sodium-potassium pumps, removing sodium chloride, okay? And water being pulled out by those higher salt concentrations. So that's what's occurring in the nephron loop. So does the nephron loop function to concentrate the filtrate?
No.
Good. What structure in the kidney concentrates the filtrate? Collecting duct. Remember, notice I didn't say what part of the nephron. The collecting duct is not an actual part of the nephron. Okay, the nephron actually ends right here. Now we move into the distal convoluted tubule. And I haven't drawn this anatomically correct. This should actually be over here. but I wanted to stretch out where I could write this material, so, well, you know, I do recognize that it should be on that side of the board. Here we do, again, primarily reabsorption of sodium ions. Potassium is affected as well, but I'll let you read about that. And this, at this site, sodium ions are going to be reabsorbed in both locations. The distal convoluted tubule location is limited to regulation. So 80% of sodium ions are removed here. More sodium ions are going to be pumped out to form the gradient. All right, and we have a hypoosmotic filtrate passing through here. But if the body needs additional blood volume, then aldosterone will be released, again, under the influence of our juxtaglomerular apparatus, angiotensin 2. And it's most effective here on this distal end, but aldosterone is the hormone of our distal convoluted tubule. ADH can bind there as well. All right, ADH is primarily the collecting duct in the cortex. Aldosterone is primarily the factor in the distal tubule. So it is, we'll have slides about that in just a moment, but it is a hormone that allows for the addition of sodium channels into the cell. So the primary effect of aldosterone is the movement of sodium from the filtrate back to peritubular blood, because our convoluted tubules, remember, are in the cortex. So I've had written here, sodium is reabsorbed and water follows. Does that change the tonicity? Does that change the osmolality of either the filtrate or the blood? If we think of sodium as our primary osmotic particle. No. Okay? It's like drinking, you know, well, Gatorade is actually even hypoosmotic to your blood, but it's like drinking Gatorade as opposed to just drinking plain water. You're taking in the electrolytes. It's supposed to be closer to your own fluid composition. So bringing in that water is going to increase blood volume and decrease urine volume. All right, so yeah, your blood pressure will go up, but the osmolality should stay the same for both. All right, it shouldn't concentrate, shouldn't dilute the blood volume. Urine, the urea might get concentrated, okay, but the salt won't get concentrated. Now, our dilute hypoosmotic filtrate flows into the collecting duct. Not much changes in the cortex. Just more and more filtrate is added. But once the filtrate passes into the medulla, now we have the ability to change the effect of this surrounding gradient. Remember, it's getting saltier and saltier and saltier as we move towards the tip of the medulla. And if the body needs more of that fluid duct, remember this is where ADH is going to bind. What organ synthesizes ADH? Heart. Excellent. I was hoping you wouldn't say posterior pituitary, but I was going to be surprised if I heard it. It's released by the posterior pituitary, okay? Synthesized by the hypothalamus. Guess where the primary site for osmoreceptors are? Receptors that are sensitive to sodium ion levels. Hypothalamus. Okay? So if you're dehydrated or you have too much salt in your blood, even though you have the normal volume of blood and the salt levels are too high, those osmoreceptors are triggered. ADH is released, and it functions by inserting aquaporins, which are water channels. It's just a fancy word for water channel. Only water can pass through. So now only water is going to be removed from the collecting duct, not water and salt. So that's going to increase blood volume, and that's going to decrease urine volume. If we add just plain water to the blood, we're going to dilute out the salt that's already there. And if we remove water from the urine, from the filtrate, without removing salts as well, we're going to rapidly increase its osmolality. Okay? So aldosterone, while it changes volume, doesn't have as much of an effect on the concentrations of salts. ADH will decrease the concentration in the blood, increase the concentration in the filtrate, because we're just moving water. Okay? So that was kind of the review, and we'll look at some slides for ADH and aldosterone as well.
Professor?
Yes?
This is decreased osmolality of blood and increased osmolality
Of the urine. Because we're adding water to the blood, so that's going to dilute the blood out. And we're removing water from the filtrate, that's going to make it more concentrated. Okay? Over here, we added water to the blood, but we also added salt. So that's why the aldosterone doesn't change the osmolality. Now, so we have adrenatritic peptide working here. We've got ADH, aldosterone and some ADH working here, ADH and some aldosterone working here. As you'll see on your lecture exam, there's another hormone that's involved. The kidneys are highly involved with several different ions. The kidneys actually make a hormone called calcitriol, which is in the activation of vitamin D and calcium uptake. And with that, the kidney, proximal convoluted tubule, and to some degree distal are affected by parathyroid hormone. So the overall metabolic activities, filtration, the chemistry that goes on in the kidney, I think is more complicated than any other area in the body. We've got pH and ion control and blood pressure control and all of that going on. Okay, so we're just kind of rubbing the fog off of a little bit, but not nearly seeing into the depth. The other information I have on there is various types of diuretics. So just as a group, what would a diuretic's function be?
Increase urine production.
Pardon?
Increase urine production.
Increase urine production. So we have our hormone, anti-diuretic hormone, which decreases urine production. All right, there's several different ways of doing this. So on the lecture exam, I have you kind of identify some of the hormones I've listed diuretics I've listed as to where they work. Proximal convoluted tubule, nephron, and part of the distal tubule are affected by osmotic diuretics. What do you think we mean by that? Somebody notice the words I have written here, glucose and mannitol, which is a form of a simple carbohydrate. So if someone is a diabetic, diabetes mellitus, and they have excess levels of glucose in their blood that can't be transported back by the symporters, it stays in the filtrate. That has the osmotic effect of keeping water with it as well. Hence the polyuria, that is a sign of diabetes. So that's the osmotic diuretic. All right, having that sugar or simple starch in the filtrate keeps more water from leaving and being reabsorbed back to the body. So that's what's meant by an osmotic diuretic. Okay? Then at the nephron loop, specifically in the ascending limb of the nephron loop, we have chemicals that are known as loop diuretics. You've probably heard of Lasix or furosemide. It's a very common diuretic given to individuals with heart problems to decrease edema. So the heart doesn't have to work quite as hard. Now since it's affecting the ascending limb, what do you think it does? The purpose being to increase urine output. What do we have in our ascending limb? The pumps, exactly right. So loop diuretics stop those pumps from functioning. They interfere with the pumps. So if we're not pumping out sodium, two things are happening. Sodium is staying in the filtrate, which works like an osmotic diuretic to maintain fluid in the filtrate. All right, so the more sodium that stays in the filtrate, the more fluid is going to stay with it. And without the pumps working, our gradient is not as highly concentrated. So as fluid flows down the collecting duct, not as much, it's like putting less sugar on the strawberries, not as much fluid will be pulled out of the filtrate. Okay, so it's going to increase urine output in those different mechanisms. And then around the distal tubule, we have what are called potassium-sparing diuretics. All right, because of our regulation of sodium, potassium can be excreted or reabsorbed or secreted. And these work by keeping the potassium. Loss of potassium is, in other words, if we keep sodium, we exchange it for potassium. So there's usually, sodium-potassium pumps that move in opposite directions. And so these diuretics work by allowing, keeping the values of potassium close to normal. Because nerves and muscle don't work very well if they can't create an action potential with abnormal values of potassium. Okay, so those are, if you work in the nursing field or the health field, there's different reasons depending on the reason why the person needs a diuretic or any underlying condition that this particular type of diuretic might be used.
Can you give us an example of one of those?
Thiazides are potassium sparing as a group.
Did you say those were by the collecting duct?
The distal end of the collecting duct, or the distal end of the convoluted tubule, and then the upper collecting duct, the one that's still in the cortex. All right, so aldosterone is one of those molecules triggered by renin release. And then from the juxtaglomerular cells, remember that they themselves can increase blood pressure by causing vasoconstriction. But they can also trigger the adrenal gland to release aldosterone. So what kind of a hormone class is aldosterone? The steroid, yes. So you can see here is, it says urine, but it really should be filtrate. I didn't make this slide. Over here would be the peritubular capillary, yes.
Is this for notes?
The slide is not, but the content is under distal convoluted tubule. What page are you on? 32. So here would be the distal tubule. Here's the peritubular capillary. And aldosterone, its receptors are going to be on the base of the cell. So from the peritubular capillaries, aldosterone, since it's a steroid, is going to pass into the cell. And its receptors are going to be on the inside of the cell. And steroid hormones, remember, activate the nucleus. This takes a long process comparatively. Causes the nucleus to copy genes. And those genes are for two different proteins, for a channel, like a leaky type channel for sodium, as well as a sodium pump. And so this is how, once this is accomplished, again, primarily in the distal convoluted tubule, but a few cells in the collecting duct. And so we're moving additional sodium out of the cell, okay? Potassium is moved in, but like we saw in the proximal convoluted tubule, it's going to leak right back out again. All right? So we have this regulated. Remember, sodium ions leave at the proximal convoluted tubule as well, but they don't leave the distal tubule unless we have these ions binding to it. So we're looking at this region right here. So potassium leaves, sodium leaves, chloride leaves passively because of its negative charge, and water leaves as well, following sodium, and then potassium is reabsorbed out as well. Then the fluid can move into the collecting duct. Okay. We have a couple of different types of cells in the collecting duct. So this is also in your lecture notes, either at the bottom of that page or the next page. I think it's at the bottom. Page 36. So intercalated cells. Again, these are found in both the distal tubule and the collecting duct. And these are the cells we'll be talking about on Tuesday. All right, we'll come back and talk about intercalated. Intercalated just means inserted between. And so notice what these cells are doing. They're moving hydrogen ions and bicarbonate ions. Okay? At the distal tubule, hydrogen ions can either be secreted, be added to the filtrate, or they can be reabsorbed. As we'll see on Tuesday, in the proximal tubule, they can only be secreted. All right, and that's because of a similar barrier to hydrogen ions that we saw in the blood-brain barrier, where we have to actually convert carbon dioxide to carbonic acid. So it's the principal cells that are affected by aldosterone and ADH. So principal cells, principally, primarily in the convoluted tubule, and cells binding ADH in the collecting duct. You have this illustration in your diagram. Go back to that familiar G protein complex. This is not a steroid hormone. All right, so it works by activating a G protein and adenylate cyclase and cyclic AMP. And those enzymes that are activated via cyclic AMP are going to result in the production of aquaporins, which are not shown here. But aquaporins are special water channels that allow only water to pass through the lipid. Normally water doesn't mix well with lipid, okay? So it allows only water to pass through. The collecting duct is not the only site that we have aquaporins. So any place where we have water moving passively, so proximal convoluted tubule is going to have aquaporins. Our descending limb is going to have aquaporins. The key difference is these are not regulated. All right, they're always there. Whereas the aquaporins in the collecting duct only appear due to the influence of ADH. Okay. And since they only allow water to pass through, that's why we see the change in dilution or concentration factor, because sodium ions are not passing through that, are not passing along with that as well. All right, so let's see. Yeah, so we've talked about the hormones already. This was a question we saw with when we talked about cardiac output. So which of these molecules, angiotensin isn't really a hormone, but which of the following result in a decrease in systemic blood pressure? Which one? D. So antidiuretic hormone is going to increase water reabsorption, which is going to increase blood volume. Aldosterone does the same thing by moving sodium in. Angiotensin 2 does vasoconstriction itself, and then causes aldosterone and ADH to be released. Atrial natriuretic peptide is going to do the opposite. It's going to get rid of fluid, it's going to get rid of sodium. Okay? So it's remember, all you have to do is think about what causes the heart to release it, and it's the stretching of the atria, which means too much fluid. Okay, so the heart's going to be interested in decreasing that. So there's a whole chapter in your textbook on fluid and electrolyte balance, and we're just going to look at very limited. All right, like I said, we could probably spend 2 months just on ion regulation and calcium and magnesium and all of that. But because of its role, renal diet, someone who's in renal failure is often restricted on their dairy products. Because of the calcium, they can't eat bananas. Because of the potassium, they have to be careful of their salt intake. All right, and that's because of the involvement that the kidneys have with so much of the body's ion movement. So water is found in 2 basic locations, inside and outside. Inside the cell and outside the cell. All right, most of it is found by proportion inside the cell, but most of the blood that's not in the cell surrounds the cell. Okay? And then we also have some of it in the blood. Where else do we find extracellular fluid? What other locations where we have fluid that's not either surrounding the cell, our tissue fluid, or in our blood vessels? Pardon? Yeah, the pleural cavity. That's the one I missed this morning. The pleural and pericardial cavities. There's only a few mils in there. Your eyeball, the aqueous humor of your eyeball. Think of a couple of more classes and locations.
Blood-brain barrier?
Well, the dural sinuses, the CSF. Forgot that this morning too. Inner ear, the fluid of the semicircular canals and the utricle and sacral joint cavities. Okay, so those are very minor places, but they're in addition to our primary. Now, we're going to be looking at some of these ions with our acid-base balance, but this is most of you already know this, the most common predominant ion in extracellular fluid is sodium. Inside the cells is potassium, all right? Notice the osmotic pressure is the same, even though the ions are different, okay? So yeah, sodium ions try to get inside the cell and potassium ions try to get out of the cell, but that's dependent on their own gradient, not because particles are higher inside the cell or particles are higher outside the cell, okay? So that's going to limit the water movement. If the cells were hyperosmotic, water would be trying to get into the cell, okay, rather than sharing those particle components. Interestingly enough, bicarbonate ions we'll be talking about as part of our buffering, acid-base buffering on Tuesday. And even though phosphate is a very prominent, it's the highest inside the cell than outside, but it is the most prominent buffer in filtrate. It's definitely an extracellular fluid. Okay? Why do we need a buffer in filtrate? We haven't gotten there yet, that's Tuesday's lecture, but why do we need to buffer our filtrate?
pH maintenance.
So we help to lower the number of hydrogen ions in our body by adding them to the filtrate in the proximal and distal tubules. If we didn't have a buffer, your pH urine would be about 1. That wouldn't feel really comfortable when you went to the bathroom, okay? So these buffers act like a sponge to take the hydrogen ions out of solution, therefore more ions can come into solution. Same idea of oxygen binding to hemoglobin, more oxygen can move into the gas space, all right? And so that allows the urine to carry a lot of hydrogen ions without lowering the pH. Here's just you have this diagram in your lecture notes, but it's just a real graphic illustration. The red and blue are extracellular fluid, the yellow is intracellular. So here you can see that sodium ions are extremely high outside the cell, and this is sodium ion inside the cell, okay? Potassium ions are extremely high inside the cell. Here's potassium ions outside the cell. So it's just a quick, your diagram in your lecture notes is not in color, but at least the dark will be outside the cell and the really light ones will be inside the cell. Okay? That just shows that as a graphic. Now, here are the different ways in which we get fluid and get rid of fluid. So, our basic metabolic activity and the food we eat, and then primarily by drinking. And we lose fluid by sweating. We have fluid in the air that we breathe out. But most of the fluid loss is through urine. About 1.5 to 2 liters a day. Anybody know what the minimum amount of urine is that your kidney should be making before someone starts worrying about you? You had a catheter in place, they're going to be measuring how much urine you make if you're on an IV, and 30 mils an hour. Okay, that's an ounce. So in an 8-hour shift, you should have a minimum of 240 mils or a cup of urine when they drain your catheter bag at the end of the day, or that you feed into the cup in the tent that's called, it's a triangular, I know it's called a hat actually. Triangular bucket, if you will, that fits under the commode seat. So if these are out of balance, then you either have too much fluid in your body or not enough, all right? So overhydration can occur by drinking too much or by not peeing or sweating enough. And there are certain kids that are born with the inability to sweat, so their body temperature rises very dramatically because that's an important way of cooling the body. And it's been impressed on us. Now, I'm not a runner. My husband trains, he did the marathon a few years ago and several half marathons every year. But they set up the water table, Gatorade table. You're encouraged to drink. If you're thirsty, you aren't drinking enough type of thing. Every year, it's less than 10. A few people die during races from overhydration. But it's significant enough that paramedics are being encouraged, if they pick up somebody on the racetrack, not just automatically start an IV on the assumption that they're dehydrated. But to wait till they get to the emergency room, you can draw blood and do a test and make sure that they're not overhydrated, because if they're overhydrated and you add an IV, you're just compounding the problem, okay? And being dehydrated, if you wait long enough to at least get the blood test done and see which way their fluids are moving. Primarily, the problem is diluting out sodium ions. And again, the effect on forming an electrical current for the muscle and for the nerves. Yes?
Yeah, that happens a lot of raves when people take ecstasy. They appear just incredibly drunk. The ecstasy, they cannot drink enough water. They just keep drinking, drinking. And then they get to the point where it's overhydration. They appear like they're drinking whiskey.
Because of the water toxicity?
Yeah, like you see people pass out that way.
There are actually people that have psychological disorders that are addicted to water. They have to be watched because they'll go into the bathroom and take a shower and just stand there and drink and drink and drink and drink and drink. So the same type of thing occurs. My father-in-law, missing most of one lung, he gets pneumonia easily, so whenever he gets a fever, his wife takes him to the hospital or to the doctor. And last fall, he was hospitalized with pneumonia. And my husband's sister called while we were on the road and said that Dad had SIADH. SIADH. And I'm thinking, this thing's an age, it's usually hypertension, but usually has low blood pressure. So I forgot my daughter had an internet-connected phone. I never have the opportunity to use one, so I got the only phone I could get without having to pay the fee. So I had her look it up, and ADH refers to anti-diuretic hormone. And he was producing too much ADH, which is an anti-diuretic, so he was retaining too much fluid. And his confusion, my husband would talk to him on the phone, if the TV was on or there was someone else in the room, he couldn't hold a conversation, he was too easily distracted. And what he said didn't always make sense. Everybody was, Dad's 86 and he's getting old and senile and it was pneumonia, lung cancer, other aspects of the lung can trigger oversecretion of ADH. So once they got that under control, then his acuity came back. And it was all how it was like cerebral edema with high altitude sickness, mountain sickness. Causes the fluid, the cells in the brain to swell with that extra fluid. And that's what was happening to him with the extra fluid from the ADH.
So if someone has COPD and they take in lots of fluid, would that be a problem? I mean,
Well, COPD in and of itself can put hard work on the heart because of the thinning walls of the alveoli. You can get the pulmonary edema, so you have to be careful with that. And the type of fluid that's being taken. Well, drinking fluid is